Hi, this is Dara Stockton with USDA ARS in Hilo, Hawaii. I'm a research entomologist, and today I'm going to be sharing updates on our collaborative work on spinosad resistance in fruit flies across Hawaii, and also the latest on our research on avocado lacebug biology, ecology, and management. First, I'm happy to really report that we've completed a Hawaii-wide resistance survey for spinosad and melonfly and oriental fruit fly in the state. This is an APHIS PPA funded project, and we did this in collaboration with UH Cooperative Extension and the Chicano Lab at UH Manoa. And as part of this project, we went across all of the different major islands in Hawaii, and we sampled for these different fruit fly species, and we compared them to our lab susceptible lines to look at how they are responding to spinosad to identify where there could be potential issues with using the approaches that have been most widely adopted in Hawaii for the last 20 years or so. To do this, we used a method of live trapping the flies. So we used these containers with holes in the side, and we used a parapheromone lure. And we were able to collect wild flies in the field. So this is a photo of an infested zucchini on the left from one of our farms in Maui. And then on the right, you can see a number of oriental fruit fly in a highly infested papaya field near Keau. Overall, we were happy to see that the levels of spinosad resistance throughout Hawaii were much lower than were reported back in 2018, which found areas on Oahu in excess of 100-fold resistance. And that level of resistance is indicated on this figure by the numbers associated with all these different points on the map. The closer the number is to 1, the lower the resistance, the higher it is, the greater the resistance. So really anything above 5 we would consider a fairly resistant population or a low level of resistance. Anything above 10 is certainly an area for concern. So we found 4 main areas that we're going to continue to monitor. Oahu melon flies up in the North Shore and Waianae area were the highest levels of resistance of anywhere that we tested. Kula melon flies on Maui both showed elevations, not super high elevations, but they were still elevated. And that's consistent with the grower reports coming from that area that products like GF120 generally aren't very effective for them. On the Big Island, we found oriental fruit flies in the Hilo area with some elevations, specifically in the university area. And we also found a pocket of oriental fruit fly and melonfly spinosad resistance elevation near the national park on the Big Island. So further south, but that is in fact nearby a golf course. And so we're concerned that there are some interactions potentially with what's being sprayed down there. So areas like Kauai though and Molokai seem to be doing really well. The numbers are great coming out of these islands. I don't think there's any reason to be concerned there. So following up on this, what we're doing is we're preparing to launch a spinosad tracking website in early 2024, and this is something we're still in development. This is a screenshot of what that website will probably look like. And the idea is we want to launch a citizen science program. So we're going to create kits that you can order or request or pick up from your local extension office with clear instructions on how to test your flies. And within 24 hours, you will get an idea of whether or not the flies on your farm do have a resistance issue. And the goals for this are threefold. First, we really want to gather more widespread data on spinosad susceptibility than what we can collect on our own. It's very time intensive to do the full-scale assessments that we did for our initial survey. So this type of approach will allow us to collect a much wider amount of data by having the growers themselves be involved in that data collection process. Second, we wanted to develop a method of rapid screening, not just for this system alone, but something that could be applied to other systems as well. We know that there's issues with insecticide resistance in other systems like diamondback moth or western flower thrips in floricultural systems. So We're hoping that if we can come up with a system that works for Hawaii for this problem, that we can apply it to other systems as well. And the last thing is empowering growers to make informed decisions. Unfortunately, observations alone necessarily of how well a product is working on their farm, without having something concrete to compare your results with, can make it difficult to know what you're really working with. So we wanted to come up with something that will allow people to make better, more informed decisions about how they want to proceed in managing pests on their farm. So it would look something like this. You'd get a kit, you'd collect your flies using a provided trap, you'd follow the instructions. Each kit will come with a few vials, and the vials are pre-coated with the toxicant. And you'll be given instructions on how to make your evaluations. Right now we're looking at about a 6-hour time window. And then once you have your results and you know how many have died, you can upload your results online. And there's also going to be a rubric for how you can interpret the results that you get. Meaning, depending on the number that die in the vial, what does that mean, both in terms of resistance level and what do you do? So although that this system is not live yet, we're wanting to get the word out that here in the next 4 to 6 months, we're hoping that this will be available. And when it is, you'll be able to go and request a kit online or pick one up from your local extension office, and you'll have everything you need to test flies on your farm. And we will make sure to get the word out when that is ready to happen. The next big thing that I want to share from our lab is some of the exciting results that have come back from our work with avocado lacebug, ALB. It's continuing, and there's been exciting progress led by Mike Bosch, who is the postdoc in my lab, who's been doing all of this work, and he's fantastic. And so he's prepared a few slides for us to share some updates on some of the work he's been doing. So first, he wanted me to share about cultivar differences in ALB. So I think some of you know because we've worked on your farms, but we've been really interested in understanding whether this newer pest in Hawaii shows clear differences in how it infests different cultivars in Hawaii. So we focused a lot on Charwell and Malama, but we've also done some pretty intensive assessments across a battery of upwards of 24 different varieties that are widely grown. Things like Nishikawa and let's see, Kahalu'us, Green Gold, etc. So long and short, his data at this point show that there aren't major differences in ALB abundance, especially if you look longitudinally, so throughout the year. Different cultivars can have different numbers at different times of the year, and it's mostly related to tree phenology. However, that said, one of the cultivars that does show lower abundances very consistently is Malama, which is consistent with what we've heard from growers as well, reporting that their Malama trees seem to be hit less hard. Leaf sugars, according to what he's been doing in the lab, Mike, is showing that leaf sugars appear to be positively correlated with nymph abundance on the leaf. So that's what this figure on the lower right shows. But there's other physical characteristics that we looked at, like thickness and toughness, and trying to just understand why the bugs were favoring some trees over others. And we found that those factors don't necessarily adequately predict movement of the pest within the crop, but sugars do. Another part of his work has been trying to gather data on the population dynamics of this pest in the state. And the data are pretty clear now. I think we're moving into we're wrapping up our 3rd year of working with this pest. And there's certainly these seasonal fluctuations. So our peaks tend to be in the late fall, followed by a sharp decline in the spring and a slow rebound as you work your way up to fall. And this seems to correlate really strongly with the leaf sugar patterns that follow that type of seasonal effect. So at this point, what we see is that the numbers gradually increase over the summer, leaves mature, reproduction picks up by this pest, and following fruiting and major production, the trees start to go into a big flowering cycle, which is when they go through a big defoliation event typically. And that defoliation essentially stops all reproduction by this pest because they can only reproduce on mature leaves. And it takes a while before those leaves mature fully enough that the populations can start to build again. He's also found there's some interesting differences at based on altitude. So because so many of our farms occur at a fairly wide range of altitude as you move up the Mana, we wanted to look at whether or not certain farms, depending on elevation, were more or less at risk. And essentially what we found is that at higher elevations, leaf sugars are more stable, and that this is likely a temperature-driven effect. So at cooler temperatures, sugar is more sustained, populations are a little bit more sustained. And so generally throughout the year, you're going to have to manage those populations a little bit more intensively consistently compared to lower elevation farms where you're going to have a much steeper crash in the spring and a little bit of a reprieve from this pest. The last big update that I want to share is that we're working really hard to put together a Western SARE grant this year. We've been invited for a full proposal, and so we're going to be getting that submitted next month. And the goal here was to understand the association between ALB-induced leaf damage and yield loss. This has been probably the biggest question that we have in terms of developing a management strategy, because at this point there are no hard data on how infestation levels ultimately affect yield in anything more than an anecdotal manner. So we want to systematically address this and try to understand, based on an economic model, what is the best time to intervene with this pest, or if at all. So to prepare for that, Mike has been collecting some data on how nymph abundance creates these chlorotic and necrotic blotches on the leaves, and we're hoping that we can develop a system in which you could do assessments on your farm and look at blotch size on your leaf, and we're hoping we can come up with a threshold value, and beyond that you can know when to engage with management. So with that, I want to thank you, and special thanks to the Chicana Lab, who we worked really closely with on all of these projects, UHC-TAR Cooperative Extension, and then of course APHIS PPA for their funding. If you have any questions, you can email me at dara.stockton@usda.gov, and feel free to follow me on Twitter. Thank you.